Olá, gente, boa tarde e bem-vindos a mais um Mock Class em parceria aqui com a SC e a EFIG Santiago Canyon College. Hoje, na Mock de hoje, guys, pessoal, vocês vão estar aqui com a professora Dr. Denise Foley. Ela é uma professora da parte de saúde, da parte de ciências, e ela vai dar um mock classes aí, ela vai explorar o laboratório da universidade, o que é fantástico. E ela vai falar um pouquinho com vocês aí sobre essa parte de vacina, sobre essa parte de biologia molecular. Eu espero que vocês aproveitem muito essa oportunidade de novo, pessoal. Aproveitem essas oportunidades. É uma professora norte-americana que está aqui hoje, de um college da Califórnia. Então, perguntem, façam perguntas, interajam com a professora. Quando a professora permitir, abra o microfone e fale com ela. Se vocês tiverem qualquer dúvidas ou vocês estão aí com vergonha de falar, o que não precisa ter vergonha, por favor, digite no chat para mim e aí eu faço essa pergunta para a Dr. Foley, tá bom? Então, câmera desligada, microfone desligado, a não ser que vocês tenham dúvidas e a professora pergunte e vocês abram, tá bom? Então, agora eu vou passar para a professora e ela vai dar essa aula maravilhosa para vocês. E depois a Jetsa e a Danielle vai falar um pouquinho, tá bom? Hello, Dr. Foley. Thank you for joining us.
Thank you. Hi. Welcome, everybody. Thanks for joining us today. I'm currently in my laboratory at Santiago Canyon College, where we teach microbiology and some of the biotech classes here. And before I get started in my class, I just wanted to make sure I welcomed you all. And I wanted to maybe take a moment and show you a little bit about the laboratory that I'm in. Since I'm here today, I thought I would show you how the laboratory is set up and a little bit about our building today. So I'm going to switch my camera right now to show the back. And then I'm going to walk around for a moment and show you what we have in this laboratory. So this is the room that I teach in most days. So a couple days a week, I'm in this lab. This is a building that is probably about 7 years old now. And you can see the stations that the students work in. Right now I have one open so you can see that the students have their own individual microscope. There's 32 microscopes in this room. And there's space for 32 students to work at and during one laboratory period. I'm going to walk around and show you a couple pieces of equipment that we have out. So right here we have some very fancy pieces of equipment. This is a routine spectrophotometer. You may have used this if you've taken a science class. You can measure the concentration of various substances in liquid. So this is a fairly normal one. I don't know if you can see inside, that's where you put the little cuvettes to measure. And this is a much more fancy spectrophotometer, and it is designed to measure the concentration in a tiny dot of liquid that you would put there. So this is a very advanced piece of equipment that some of our students get to learn how to use. I'm just gonna walk a little bit around and show you how the room's set up. So these are some of the incubators that we set at different temperatures in order to do some of our experiments. This piece of equipment right here is a fast performance liquid chromatography. It's a very expensive and advanced piece of equipment that the students that are taking the protein class would learn how to use to purify out proteins. And over here are a couple of our robots that we showed students how to use. There's an automatic diluter right here. And I'll show you this working in a little film during my presentation. It's an automatic plate pourer. So we can make media plates that are very precise and kind of industry grade. I'm gonna walk into the prep room just so you can see. This is where some of our student assistants would work along with our science coordinator to prep the materials for our class. You can see there's a biosafety hood and some of our supplies in here. And Right now, there's not too much going on because of the COVID-19 situation. We're not currently having classes on campus, but this is where we work. That was just a little bottle of glass cleaner. Okay, so this is the room that we work in. I'm going to change my camera back around. That's an incinerator that the students in microbiology use. We'll plug this back in, and let me change this back. And here I am. So this is the laboratory that I teach my class in, and I need to adjust this. I'm sorry. Okay. So I'm going to try to share my screen right now so that I can show you my presentation. Oops, I need Diego. I can't share my screen, so I can't get my presentation. Can you allow me to share my screen?
Please try again.
Okay. There we go. Okay, so today we're going to talk a little bit about DNA. And if you've taken a biology class already, some of this might be a little bit of a review for you, but hopefully I'll teach you a little bit about an application that perhaps you haven't learned about. And we'll try to keep it at a pretty basic level. But if you have any questions, be sure to send them to Diego via the chat, or you can ask me as we go along. I'm happy to answer questions for you as we move through this. And then I'll also be interspersing a little bit of information about our certificates and the degrees that we have here in biotech at the college, and how that relates to some of the careers that are out there. Okay, so that's my information on this slide. See if I can move this. This might be. Okay, so this is the information for me. I am a department co-chair and the program coordinator for biotechnology here at Santiago Canyon College. I've been teaching here full-time for 14 years, but before that I was teaching full-time for at least 9 years. That tells you how long I've been teaching. I love my job, and I hope that the students get a lot out of my classes. I think they do. All right, so here we go. Who are we? This is me, right? So you've already been introduced to me, Denise Foley. This is my colleague, Anson Louis. He also teaches full-time in our biotechnology program, and he also teaches the cell and molecular biology class for the majors. This is Novi Boss, another full-time professor in the biology department, and teaches the introduction to biotech classes. And also he's a nucleic acid expert, worked in the industry for a number of years, and brings his expertise to our campus, and we love him. And this is Danielle Alared. She's on the call today. She's our career coach for biotechnology. So she works a lot with high school students and helping them navigate entering into our program here at Santiago Canyon College. And she's just very phenomenal. She helps us with so many things in our campus. We love her. Okay, so what is biotech? Hopefully you have a little bit of a vague sense of what it is, but often students don't really know what the definition is. And so this is a fairly simple, straightforward definition. Biotechnology is the application of science and technology to solve problems and make useful products. It tends to use living cells to do this. So we tend to work in the realm of things like proteins and DNA that come from living cells, and living cells produce these things for us in order to do the things that we desire to do, right? What we want to accomplish. There are a lot of opportunities in biotech. This field spans very many different applications. So you can see just a few of them on this slide. This field is huge and it has lots of opportunities. So if you're interested in a lot of these different areas, training in biotech will help you enter into these careers and move forward. So we'll touch on a couple of things today, but some of the things have been in the news recently or in the past, right? Things like vaccines and new drugs to treat this virus that is causing this pandemic, right? Insulin, you might know somebody that needs insulin to treat their diabetes. And insulin was the first genetically engineered biotech product that was brought to the market. We'll talk a moment about using DNA to test paternity. When we do our exercise today in the class, you might be able to imagine how that exercise extends to determine things like paternity for organisms and for people. It helps with developing new food products like food science, new agricultural products, ensures things like clean drinking water and a sustainable fuel supply by developing new biofuels. Those are just some of the things. There's lots and lots of different types of fields and careers that rely on the techniques and tools of biotech to accomplish what they want to accomplish. So let's talk just a little bit about some of the important sort of facts about DNA in order to introduce this important molecule today into our class. So who knows? Hopefully you already knew that there are 4 different what we call letters in the DNA alphabet, but these letters really are referring to the nucleotides that make up the molecule, right? The A stands for adenine, the C cytosine, the G for guanine, and the T for thymine. You probably knew that. And that's some of the basic information that you learn when you start to learn about this DNA molecule. But we need to kind of remember these letters as we go through this exercise today. Well, here's the next question or statement. The human genome, right? That's our genetic code. And that's what's required to create a human being. Do you know how many genes there are in a human being? Well, you might be taking a guess right now. I'm gonna tell you that they think, oh, so do we have some chat answers? Let's see, does somebody have
Lots.
lots? Okay, what are they saying?
Yeah, so Camila said lots. And please, guys, have a guess. Open your microphone and talk to Professor Foley. It's going to be a great opportunity. Please, guys.
Yeah, let's have a few guesses.
So I don't know exactly how much we have, but I suppose there must be more than 1 million.
More than 1 million?
Yeah.
That's a little bit high. So someone take that's a little bit lower.
Okay. 40,000?
48,000, you're closer. You're definitely closer. And there was a time that they thought that it was about that, but they've actually brought it down a little bit lower than that.
Okay, 30,000?
Oh, now you're about right. Okay, so they think that there's probably somewhere between 20 and 25,000 active genes, but there are more than that are probably not completely active, and we haven't really figured it all out yet. Okay, so they don't know everything, but they think that there's about 20 to 25,000 active genes, and there's a lot of other genes that are in there that aren't really active right now. So let me see if I can go. Oops, I'm stuck. See if I can get to the next one. So the Human Genome Project, maybe you've heard a little bit about that in the news. It's been, a number of years now that they finished sequencing the first human genome, right? All of the DNA that were in a human. So the first person's DNA to be sequenced, that took about 13 years to figure out, right? There was many different scientists all over the world that were working on this set of DNA, trying to piece it all together and determine the order of the letters in the genome. And as I mentioned, it took 13 years and over $1 billion went into that research to decipher that. Does anyone have any idea how much it costs now? Just take a guess. If you wanted to sequence somebody's genome, how much would it cost and how long would it take nowadays?
Nowadays? Okay, I'm probably gonna commit a mistake, but I suppose would be 50,000 and maybe 2 months.
2 months.
I don't
Okay.
know.
It's actually gotten even faster than that. Good guess. Just like any technology, things get better and faster. If you think about, the first computers were about the size of a room. And nowadays we have a more powerful computer than that first computer in our pocket if you're carrying around a phone, right? So things get faster and better. So now if we wanted to sequence the entire genome from start to finish, we could do it in a day and for a few $1,000s. So that's a huge difference than the first one, right? So we've gotten better at that. So there are a lot of letters in the human genome. If you take all of those letters, G, A, T, and C, let's try 3 guesses. Who wants to take a guess? How many letters are in the human genome?
I think that are hundreds of letters.
So there's 4 letters of it. How many? You think there's a few hundred of them? But remember, there's already 25,000 genes. So you think you need more than a few hundred?
Oh yeah. So thousands.
So there's thousands. Okay, let's take another guess. Who wants to guess? We have thousands.
Okay, I don't gonna answer that one because I don't really know.
You don't know. Does somebody else want to take a guess?
Sorry, Professor Paola. She said billions.
Millions? Okay.
No, billions.
Billions. Okay, so how many billion? That's actually a pretty good guess. How many billion? Somebody give a number.
3 billions?
Yeah, you're right. Actually, it's over 3 billion letters. So that is a lot of letters in the human genome. So if you would write it out, if they're going to write it out, how much would it stretch? Well, if you took these letters and you typed them all out, it's going to take 9,000 kilometers in order to write all of those letters out. So I know 3 billion is just a number, right? If you try to imagine how many numbers that is, it's going to take you 9,000 kilometers to finish all those letters. That's a lot. And so in miles for us United States people, right? We think more in miles, even though the rest of the world is in kilometers. It's about 5,500, almost 5,600 miles. That is a lot of length to that, typing that out. And talking about typing, if you took a typist and you said, okay, type out that 9,000 kilometers of letters, right? The 3 billion plus letters. Somebody throw out one guess. How long do you think it would take a typist working 8 hours a day to type out that genome?
Okay, so with our technology of nowadays, I think maybe a week.
A week?
Yeah, I'm really not good at this.
So in a week, somebody could type out 9,000 kilometers of letters. Actually, that's underestimation, because if somebody was working, a typist was typing 8 hours a day, they would take 50 plus years to type out that genome. I'm just giving you this information so that you can think about just how enormous our genome is, a molecule, right? It's huge. It's very, very big. So it would take over 1 million typed pages to get that genome typed out. And if you stack those pages up, it would be 200 feet high. So that is a lot of letters in our genome. And so we're going to be moving into our topic about DNA in just a moment.
Sorry, Professor, before you go further, Mateus Faria Soares, he's asking, how can cell support this number of information?
Well, so the molecule is actually very, very small, right? And so there are proteins that help organize that DNA molecule. They're called histones, and they wrap the molecule around them, and then they twist even more. So they're very, very organized within a cell, and it's all crammed within the nucleus of the cell, and it's highly regulated. You would learn more about that in some of your biology classes. So it is mind-boggling to think that this tremendous molecule, this huge, long, molecule with all this information is packed inside our cells. But you have to remember the scale, that this molecule is actually, very, very tiny. And so we're able to organize it and pack it into a cell. But yes, I agree. It is very hard to imagine that this is possible, but it is. Our bodies are very, very amazing creatures. Is there any other questions that I can answer? Okay. So I'm just going to show you a little bit about the equipment. I already walked around this room a little bit, but I wanted to reiterate to all of the students on the call today that if you came to Santiago Canyon College, you would be being trained on equipment that is used in industry, right? It's industry grade. This isn't, sort of hokey pokey kind of equipment. This is the real deal. And so you'll see a student up here that's working with a micropipette. You'd become very efficient. Excuse me. You become very proficient at using that. And also the microscope, excuse me.
So guys, SCC, it's a great college in California. And as Dr. Foley was saying, it is very directed for the industry. And you guys, when you get in this, for example, this laboratory, you'll be in touch with all those amazing equipments that is going to prepare yourselves for the industry, right, Professor?
Yes, thank you. Sorry, I have asthma, and sometimes I have a little coughing fit when I start talking. My apologies.
Don't worry at all, please.
Okay. Drink some water, maybe it would help.
I did. Thank you. I did. I took a little sip. Stepped out for a second. So I just wanted to point out some of this equipment. This is all equipment in our laboratory. These are our students that are working on this equipment. So we have a pH meter, student working with it. This is a gel analyzer machine for the DNA and the protein work. Micropipette I mentioned. This is a special microscope that's in the tissue culture room. We have a class on teaching students on how to work with cells and culture. And so those are those equipment. This next slide shows you just some of the things that we have here. I'm not going to list all of these, but these are different pieces of equipment that the students learn how to use and how to work with in the lab and do different techniques with them. So this is not even a full list, but just a kind of a overview of all of the different things that students can learn how to use. And as I mentioned, this is the robot of the plate pourer, and it's making some plates right now. It's in action, so you can see that working. On the other panel, we have students working with our DNA sequencer. This is one of our, well, both of these are newer items that we have in our program. The DNA sequencer is probably the newest, and because we have an expert on nucleic acids, that professor is able to show students how to sequence DNA and actually determine the order of letters on a given piece of DNA, which is quite amazing. And I should reiterate that these items of equipment are often better than, well, they are better than most universities have in their labs. And most of the time, if the research departments have some of these, they advanced students or something like that. But our students are trained right from the beginning how to use all of this, and that opens the door for the students to work in industry. All right, we're about to move into the class portion. And what I wanted to talk about for a couple of minutes before we enter into the sort of mystery for you all to figure out, the little case, is that DNA molecule is able to reveal relationships. And we're going to use a part of that information to a sort of like a little sidebar of that to help us figure out a case. So when you look at a gene, right, a segment of instructions in DNA, you can see that there's a sequence. And if we compare that same gene amongst different animals, organisms, people, we'll find some differences in that gene. And sometimes, right? And that helps us figure out who's most closely related, which are called phylogenetic trees. That name isn't so important right now, but just the graphical representation, I'll explain for a second. So if you look out here, the letters are just individuals. That could be different organisms, different people, different animals, right? So these are the organisms, the living things that are of interest that are being compared to each other. And what the tree is telling us is that if we look at this, it says like, well, A and B are more closely related than they are to C and D, but C and D are more closely related. And all four of these, A through D, are more closely related than E is. So if you kind of imagine an analogy like a family, then A and B are probably siblings, and C and D would be their first cousins, and E would be a more distant cousin. But if you're looking at organisms, you can just kind of see who's more closely related to who, right? So if you go back, further back into the branch points, you would say like, oh, well, these might be grandparents and great grandparents as you're going back. That's just giving you an analogy when you're trying to figure out who's related to who by looking at the DNA sequence. So keep this sequence in mind. This is just an imaginary section of a gene. And on this next slide, you'll see that if we're comparing this top one, right, this first comparison organism has a single difference in the sequence. And then the next organism has two differences compared to that, comparing that sort of ancestor organism or the comparison organism. And then the organism on the bottom has a lot of differences compared to the first. And keep in mind, we would be looking at the same area of the genome, right? Maybe the same gene in the genome. Well, these types of differences allow us to build these kinds of relational trees. And maybe in biology class, you talked a little bit about the domains of life. This was the first kind of tree that was built from these kinds of sequences. And they realized when you looked at it that, you know, we had three huge groups of life. We had the eukaryotes like ourselves, right, that have DNA inside our cells, inside a nucleus. And then we had the bacteria, and then we had another type of prokaryotes, the archaea. So life was split up into these three giant groups based on the DNA sequence. And if we look over here, we can see that like all the animals got grouped together, all the fungi got grouped together, all the plants got grouped together, right? So these groups are more closely related to, you know, other organisms in their group than they are to each other. But all of the organisms in this eukaryotic group are much more related than the bacteria. Okay, I see a lot of chats might be coming up, a little notification. Diego, is there something that I can answer for some people?
Excuse me, can I make a question?
Sure.
Cloned beings have the same gene?
Do cloned beings have the same gene? Yes.
Oh, like exactly the same, all the same letters?
They would have the same letters, and so would identical twins.
Oh.
So at the genetic level, identical twins and cloned organisms would have the same. But your brother and sister, your mom and dad would have similar, right? And some things are the same, but not everything's the same.
All right, thank you.
Mm-hmm. And every generation, right? So every generation will accumulate a few more changes that are random, right? Random mutations that occur. And oftentimes they don't have an effect at all. Sometimes they're detrimental, right? Depending on where in the genome they're occurring. Okay.
Excuse me, can I ask a question?
Sure.
Are the genes like linked to our personalities?
Well, there's a lot of work that's being done. That's not my area of expertise, but they are trying to figure out if certain variants called alleles, right? So certain varieties of specific genes tend to make people more or less different ways, right? They tend to do things like study mice, and they might find that, a mouse with a particular variant tends to be more bold, right? More daring, and try to investigate new areas more so than another mouse that might be a little more cautious and timid. But that work is still early. They don't have a lot of information, but they do have some. So it looks like some variants of genes can influence our personality, but there's probably very many things that influence a person's personality. And a person's personality can kind of change a little bit over time, right? You're not completely static. If I think of myself back when I was a kid, I'm not exactly the same as a kid as I am now. All right. There's certain things that kind of follow me through, but I'm definitely not exactly the same type of person I was then. So I think there are probably a lot of things that affect our personality.
Sorry, Professor, we do have a question here. Isadora Zani, she's asking, how the machines that you show can read a DNA sequence?
Okay, so that's a process that would probably take a whole lecture or more in itself. But in essence, the students purify out a very specific portion of a DNA, and then the machine is able to actually read nucleotide by nucleotide which letters in which order. So it is a process. You have to go through a very specific protocol and use very specific reagents, but it is definitely possible. And you would learn how to do it if you came and take that class. Okay, can I go on, or is there something else?
No, you can go for it, Professor.
Okay, so I'm just gonna show brief, since a virus is very much a part of our consciousness right now, right, with the pandemic that is going on. I wanted to show you that they use that same type of analysis to look at relationships of genes in viruses. And I found this comparison between the flu virus and the HIV virus. I couldn't find something that was so similar with the COVID virus yet, probably because it's still kind of new and there isn't a lot of information out yet, although I'm sure some people have it already. What I wanted to show you is if you're building those relationship trees, you can see that the influenza virus and the HIV virus is different. And the reason why I wanted to bring this to your attention is that you probably know that you would have to get a new flu vaccine every year, right? And that's because the flu vaccine, the flu virus changes so much from year to year that you need a new vaccine. And so if you built a tree, and these are all relative to each other. I took this from this publication here, right? So this influenza virus, if you built a tree, you can see that it has this much change in the world in 1 year. Yet this is enough change that would require us to have a new vaccine, right? We need a new vaccine in order to fight influenza. If you look at one individual that was infected with HIV and look at all of their viruses that they have after 6 years, it looks kind of like influenza. And this is just in one person, right? So they look kind of similar. And then if you look at one country in 1 year, you can see how much more change there is. And in the Congo, where they think that HIV actually jumped from chimpanzees to humans first and then spread, you can see how much variation there is in the viruses that are there in 1 year. So while we can deal, at least to some extent, with the variation in influenza and develop a vaccine every year, this much variation down here with HIV has prevented us from coming up with a good vaccine. Now the COVID virus, right, the SARS-CoV-2 virus that causes COVID is not mutating as fast as influenza. So it looks more hopeful, right? We to come up with a vaccine that actually works for that virus. So that's just kind of a side note, but I wanted to show you how relationships are built, right? How they compare one organism to another or one virus to another to show how they're related to each other. And we're gonna keep that in mind.
Professor, can I make you a question?
Yes.
Talking about COVID, you know about a kind of a evolution of COVID that it is in France, if I'm not wrong. I just listened about it. Do you know something about?
Well, I'm not an expert in the virus, but I do know that the virus is changing, right? But it's not changing at a rate that's as fast as influenza. So it still looks hopeful, right? We do know that the virus is mutating and changing a little bit, but it's not, it doesn't seem to be so fast that they wouldn't be able to come up with a vaccine that's protective. So that's the hope, right? We haven't done it yet. The scientists have not figured out a vaccine yet, but they're testing some candidates. So hopefully they'll work. So I don't know if that answered your question, but it is changing.
Thank you.
All right, so we're going to now use the DNA to determine the identity of somebody. And we're gonna use a method where we don't have to sequence the whole gene. That would take too much time. We're gonna use kind of a little bit of a shortcut, but keep in mind that those changes have occurred at the DNA level, right? That there are variations at that nucleotide or the letter level, that they're not all the same person to person. And we're going to use those differences in order to determine whose DNA is who. So this is a very, very simplistic overview of the technique that we're going to use. So the technique is called making a DNA fingerprint. And the reason why it's called a fingerprint is because you might know that your fingerprint is supposed to be unique to you, right? Nobody else has your exact fingerprint. Similarly, if you look at specific areas of your DNA, you're the only person that has that specific sequence in that specific spot, right? Most of our DNA is identical to each other, right? That's what makes us human. But there'll be certain regions that have some differences, and we're gonna hone in on those differences. Those are the areas that have been studied by specific scientists, and then specific procedures have been developed in order to be able to kind of exploit those differences at the gene level to figure out whose DNA we're looking at. So this is the overview. First, we'd have to get DNA from somebody, right? And your DNA is in all of your new, so white blood cells have it, doesn't, but your white blood cells would have it. So you can find DNA in blood or some cheek cells or skin cells, or even at the base of your hair shaft where that little root is attached. All of those things have DNA attached to them. So we have to get a little bit of DNA from a person. We're gonna copy the important region, right? We don't want the 3 billion letters. That's too much to deal with. We only want a little region to be copied so that we have lots of copies of a specific little area. It's kind of like thinking of, well, if we had that, that pile of pages 200 feet high, we're just gonna take one page and copy it and make many copies of that one page, all right? So we want to copy an important region. We're gonna cut the DNA into fragments, and we'll talk about how we're gonna do that. And then we're gonna run the DNA out on a gel and look at the patterns that result from the fragments. So this is the overview of what we're gonna do, and we're gonna go through it right now. So hopefully I can get through it in the next few minutes. So the process of making a copy of DNA is called PCR, or polymerase chain reaction. So what this process does is we're going to take a gene of interest, right? Not the whole genome, but one little section of the genome. And we're going to make a copy of that. And in order to make a copy, what we need are a few things, which are shown kind of in this panel over here on the left. So we need the nucleotides, right? That's what's said here, the deoxyribonucleotide triphosphate. So we need the Ds, the Gs, the As, the Ts, and the Cs. That's what this is. We need some little short single-stranded pieces of DNA that are called primers. They're going to be complementary to the edges of the parts that we want to copy. And we need the enzyme, DNA polymerase, that's able to make the copies. Now, in this process, we're going to take the polymerase from a bacteria that happens to be heat stable so that this protein won't be destroyed when we heat it up. But the process is just going to repeat 3 steps over and over and over again, okay? The first step is to heat up the DNA, right? So this is our template, the original DNA that we want to copy. So it could have come from blood or skin cell or anywhere that had DNA. We only need 1 copy to start with, actually. We don't even need very many copies. We just need this 1. We're going to heat that double-stranded DNA up, right, to 94 degrees Celsius, close to boiling, in order to separate the strands from each other so that they're now single strands. Now, in the tube, we have our nucleotides, we have the primer, and we have the polymerase. So after it's heated up, we cool it down a little bit to about 65 degrees. And that's going to allow, or less, right? It's going to be specific to the primers. We're going to cool it down though, and the primers now are going to stick to the complementary regions of DNA. So you'll see it stuck here and here. And then you're going to heat it up a little bit to be the optimal temperature for this particular protein enzyme, the polymerase from the bacteria that can then copy the DNA. So after that first set of 3 steps, we have now 2 strands. So we started with 1, now we have 2. And then we repeat that whole 3 steps again. And so if we had 2 strands, now we have 4, and we do it again, and then we have 8, and we do it again, we have 16. You do that about 32 times. So after you're done with this, if you only started with 1, you're still going to have
Sorry, Professor. Your internet just broke down.
Now just wait.
Yeah, just 1 second, guys, please.
Let me see if I
Professor.
can plug in. I'm plugging it in to see if it comes.
Thank you, Professor.
Did that help?
A lot. Thank you.
Okay.
Thank you.
So did that idea come through though, that if we start with 1 set of double-stranded molecules, then we go through those 3 steps over and over again, and we'll end up with millions of copies of that same area of DNA? Okay, so that's our copying step, and we use PCR. So this is a very common procedure for people to use nowadays in a laboratory. And again, if you came to SCC, you would do PCR in probably several classes. We use this procedure a lot. It's a very common procedure that people use to copy DNA. The individual Carey Mullis that figured this out, he actually won the Nobel Prize for figuring out this process. It's so useful. People use it all the time. So first step, we've copied our DNA. And this is the machine, a thermocycler, that can ramp up through all of those temperatures over and over again. So we have one like this, and we have several other types of thermocyclers here. But this is the piece of machinery that allows you to put your tubes in here with all of your reactions, and then you program it so it goes through the 3 temperatures over and over again through 32 cycles or so. The next step is cutting our DNA. So we're going to cut the DNA using another enzyme called a restriction enzyme that is able to recognize a specific letter pattern. So you'll see that there's a sequence up here recognized on this sequence, and this particular restriction enzyme is going to cut it. So let me kind of zoom in on that. So you'll see the sequence right here, and the sequences that are recognized by these enzymes called restriction enzymes, and there's a lot of different ones, they tend to be what's called a palindrome. A palindrome is something that's the same forward and back. So if we're looking along this top strand here, we see that sequence A, G, C, T, T. If you read in the opposite direction along the bottom strand, it's the same, A, G, C, T, T. So that's the characteristic of a palindrome. And the enzyme will cut the DNA right here and leave a little overhang that you see like right there. So we need these restriction enzymes for this next step. And there's lots to choose from. I have just a couple of examples, more examples here, but there are literally hundreds of restriction enzymes that are available. And now you literally just go into a scientific catalog and order the one that you want, and they'll ship it to you. There's lots of different restriction enzymes, and it tells you which site they recognize, what sequence, and then how it cuts it. So it cut it on the top strand up here, and on the bottom strand it would be down here, right? So you cut the strands like this. There's lots of different ones. This is just another reiteration of that Hindi 3 and how it's cutting its sequence. All right, so what have we done so far? We've copied the piece of DNA that we wanted, and then we cut it with specific restriction enzymes. We're not done. We have 3 more. The third step is last, right? So what we're gonna do is we're gonna have to look at differences and be able to visualize them. So how we see differences is in the patterns that will be created after we cut with restriction enzymes, the same area, right, in different individuals. So we've copied the same area in different individuals, and we cut it with the same restriction enzyme, but they're not going to cut exactly the same, right? So restriction fragments have different lengths. So we call them riflips. So we use the restriction enzymes to get different fragments that are different lengths. So polymorphism means different shapes, right? Or different lengths. So if you look, I have an orange representation and a blue representation and an orange representation. These are supposed to represent 2 different individuals that have been copied, right, in our copying process, the PCR, and then we cut them with the same restriction enzyme, but they cut differently. So if you look at the top one, how many cuts would a particular restriction enzyme give you? Can you count?
5.
5 cuts or 5 fragments? It's
5
here, this cut
fragments.
Here.
Yeah.
Yes.
4
So
cuts.
Yeah, 4 cuts, but you answered the second part, how many fragments? You get 5, right? On this individual, how many cuts? Only 2.
2.
Yeah. So, but how many fragments?
3.
3. Right. So if we were to run this out, if we were able to visualize this, we would see that they looked different, right? This is what we're going to do for our third step. So the third step is to separate out these fragments and visualize them. And we use a process called gel electrophoresis. So you see a close-up picture of that right here. So the gel is made out of a substance called agarose. It feels like gelatin or jello, right? But it's slightly different chemical substance, but it has that same sort of consistency. And you can pour it when it's still melted, just like jello. And then you have sort of a little mold that we call a comb that makes these little wells, and you pull it out and you have little holes, right? Little dips, which we call wells, that you can load your sample in. So these are our samples of DNA being loaded into the gel, and you apply electrical current to it to pull the DNA through the gel. So you can see here the representation. So we have the wells up at the top, and you're going to apply the electricity. So the positive electrode is towards the bottom, the negative is towards the top, and the DNA has an overall negative charge. So it starts to move through the gel, and it's going to separate by size. The smaller things are faster, so they move through the gel faster and get to the bottom first. This image over here is a real DNA gel, and the real gel, you use a dye that will bind to the DNA, and it fluoresces when you put a specific light on it, so you can see the DNA and where they're migrating. Okay, so that's our third step.
So, Professor, just 1 second. How does this machine work? How's this chamber? So you said you got the negative sides on the bottom, right?
Mm-hmm. Yeah.
And then when you add the gel, you said the DNA, the small particles goes first because they are smaller, so it goes first to the bottom, and then the largest particles stay on the top. I didn't get it.
They go slower. So when you add your DNA here, right, and there's a buffer in here, and then you apply your electricity, then the DNA molecules start to move towards the positive electrode because they're negatively charged, but they're going to separate by size. The things that are smaller are going to go faster. They can weave through the gel faster than the big things that kind of get hung up and take longer. And so the biggest things will be on the top and the smallest things will be on the bottom. So you can see that's happening right here, right? You can actually see this gel over here that's orange. You can see that there's some big things towards the top and there's some smaller things at the bottom.
Yeah,
okay.
Thank you.
Mm-hmm.
Excuse me.
Yeah.
I have a question. If our genes are unique, like none is like similar to each other, how does it work, the paternity tests?
Yeah, no, it's not that they're nuns. We are very similar. Oh, and I'll leave you with the thought at the very end. We are extremely similar, but there are regions that are known to be more different than others. And so each person would be unique. So most of our DNA is very, very similar, but some regions are different than others. And so we focus in on the ones that are different. And the paternity test, I'm going to show you in a second. Hopefully you'll understand when we do the exercise. I'll come back to that thought and hopefully you'll understand, okay? I won't forget.
Okay, thank you.
Okay, so let's go to you now, keeping in mind the things that we've just talked about. This is our little scenario, and hopefully in the next couple of slides, I'll be able to finish so we will be close to our time. So here's our little scenario. Who stole the diamond bracelet? A jewelry store owner left his store briefly, but locked the door. To his dismay, he returned only 10 minutes later to find his door was broken and his jewelry case was smashed. The expensive diamond bracelet on display was missing, along with some earrings and a ring. The detectives arrived and collected evidence, which included a small smear of blood on one of the broken panes of glass and a video of the event. The thief wore a mask to disguise their identity. Can the case be solved? Possibly, right? If you have the right suspects to check the DNA against. So what do we have to do? We have to go through that process we just talked about. So if we have suspects, we're going to obtain their DNA, right? From a cheek swab, perhaps, or a drop of their blood. We already have blood from the crime scene because it was on the glass. So if we have suspects, though, we're going to want to get some of their DNA in order to compare. We're going to then do the PCR, and we're going to have to follow a very specific protocol that's already been worked out using very specific primers. We're then going to perform very specific restriction digests. Again, this procedure is already completely worked out, and you have to follow the exact instructions using the right enzymes. Then we're going to do gel electrophoresis. Again, we have to have the right percentage of augros and follow all of the right instructions, right? The right voltage and everything. So we're kind of just talking about it in general, but there are very specific procedures that would be followed. And then we're going to compare the patterns that result. If we have suspects, that's great. But sometimes, right, the police departments already have databases of DNA from, you know, other criminals that have been arrested in the past. And we can actually compare our pattern that we get even from the database if we don't have suspects to compare. So here's the result, right? Sort of a simplified overview. Whose DNA is at the crime? We have our crime scene DNA, right? The drop of blood on the glass that looks like this. And we have 3 suspects that we're comparing it to. Can you see who's matching?
Suspect number 2.
Exactly.
Okay.
So in real life, the gels wouldn't be, you know, quite this simple, but this is the idea. This is exactly how DNA fingerprinting works. They're gonna look at the crime scene DNA, and then they're gonna look at the suspect's DNA. And if none of these suspects' DNA matches, then it's not their blood on the glass. And keep in mind they had a video, right? So if they see there was only one person that did the break-in and broke the glass and stole the jewelry, then they know there's only one person's blood that could potentially be on that glass. And so if none of the suspects match, that gets them all off, right? None of them are the ones that perpetrated the crime. But it might be that when they run this pattern through the database, they get another hit. Maybe this is a person that was already arrested in the past and their DNA is in the database. So this is how it works. Now, for a paternity test, we go through the same type of process, but you would be comparing the child's DNA to mom's DNA and then potential fathers, right? And what will happen is that the child will share half of the DNA bands with mom and the other half with dad. So if the child has bands that do not appear in any of the other potential, you know, fathers, then none of them are the father of the child, right? So the child will have bands that match mom and bands that match dad. And so you'll be able to tell who the father is by comparing the bands between all 3, mom, dad, and child.
And always 50-50, right?
Yeah, about 50. I mean, humans are a little different, right? We have crossing over and things get a little complicated, but for the most part, yeah. And you're not going to find bands that exist in the kid that you don't find in either mom or dad.
Okay, great. Thank you.
Thank you.
Okay, so I just want to end my little class today just reminding you that there's so many different opportunities in biotech. You can do whatever interests you. So while we talked about paternity, you're going to be using tests much like the one that we just talked about, but that's also used in forensics, which is the example that we used today. But there's so many things that you can do with biotechnology. Depending on what you're interested in, you can take the tools of biotech and apply it to your future career and enter into lots of different areas depending on what you like. Okay, so this is our core course requirements. You can see that our certificates are built kind of stacking on top of each other. So we have a very entry-level certificate that basically has two biotech classes. You enroll separately, but this is one class, a lecture and a lab, an intro to biotech. Then you have a basic skills class and an intro chem class. And that gives you sort of entry-level experience. You could potentially get an internship after you just finish these classes, and then you build from there. So we have our protein specific class. You take some more chemistry, some regulations about the industry, and then at the bottom, the next level contains the nucleic acids class, a tissue culture class, and you have some classes to pick from for some electives. Okay, so I'm kind of skimming because we're running out of time, but this is what our class looks like. You would be learning in these classes lots and lots of different techniques. You can see these different techniques that are scrolling through. So as you're moving through, you're learning how to do all of these different types of things, and they're kind of being reiterated and built upon one class to another. You get a lot of hands-on training. It's not theoretical, you're really working. So this is just information on the Intro to Biotech lab class. This is a GE class, meaning that if you just took this class and then decided, nah, I don't really want to go into this field after all, it still is a GE requirement. So you've already taken care of your life science requirement to transfer to a university. But hopefully you would like to continue and you would take the next class, which is the class that you really get proficient at using the basic equipment like the micropipetters and the pH meters and the spectrophotometer and things like that, and get some of the very basic skills. And so finally, I'm going to end my class today just saying that we talked a lot about DNA and kind of focused in on the differences, but I do want to bring us back to this thought that every human shares 99.9% of the same genetic information. We are very, very much the same. We only have 0.1% difference from person to person. That's what makes us individuals. So we have way more in common than we do are different. So we should keep that in mind as we're moving forward in our life, that we are far more similar than we are different. We're all humans. And that's the end.
That's fantastic, Professor. Thank you very much. It was a great class. I just want to open for questions. So has anyone have any question?
Actually, I have.
Okay, go for it, Carolina.
In the crime scene, for example, in real life, we will see the experiments on that light you showed us.
Yes, yeah, you would.
Oh, that's cool.
Yeah,
Thank you.
Like that orange one with the band showing up.
Okay.
I have one question.
Oh,
sorry.
No problem.
Continue, please.
It's physical, not on computers.
Sure.
Okay, thank you.
Sorry, Matthews, you can go for it.
I can?
Yes.
Yes, go ahead.
Professor, I am working in research, and I can who I can do the question.
So you can just you can just tap on the chat box, and then I can read for Professor Foley. Or if you want to ask here, Matthews, Dr. Foley is listening to you.
Yeah.
I go
on the chat.
Yeah.
Okay, that's okay. No worries. In the meantime, while Matthews is typing, has anyone got more questions for Professor Foley?
I have one.
Okay, go for it, Giovanna.
About the paternity test, when they are doing, how do they know that they get that exactly DNA, that it's not the different one? Because if they get the different one, they would do the paternity test.
I don't know if I understand the question of how do they get the right DNA and not the different one?
Because if they get the different one, they will do the test with them?
I'm not sure if I understand. So if you're doing a paternity test, you might have the situation where maybe the mom's not sure who the dad is, and you're trying to figure out who the dad is. You would get DNA from, both potential fathers and compare. They do this for animals a lot, right? So for example, birds, sometimes this is how they figured out that birds aren't always just, having offspring with one potential partner, but there's multiple partners. So they take the different baby birds and they do a DNA fingerprint, and then they have the mom bird and what they think is the father bird, and they'll realize that some of the offspring don't match. So it wasn't from the the bird that's acting as the father and helping out with the nest, that the mom bird had other potential mates. So they've done that. They've shown that animals don't always just one potential mate. And the same thing is true for humans. They can figure out, looking at the pattern, if the offspring match both potential parents or not. Obviously the mom, you know, the infant or the offspring comes from the mom, but it's not always obvious sometimes who the dad is. Does that help?
Yes. Thank you.
Okay, great. Professor, I have a question from Diego Santana, and he's asking how electricity is related with DNA. What about magnetism and DNA molecules?
Well, I don't know too much about magnetism. Electricity has been used for a long time to separate out DNA. I'm sure there's probably experiments using magnets, but it's not nearly as practical. There's the equipment that we have that is available for purchase all uses electricity. So, you know, you just plug your apparatus in and it's going to work. I don't know that magnets are strong enough to give you the type of reaction that you need to, right? We want to separate all those bands and have them migrate through. So theoretically, DNA is going to behave specific, predictable ways with magnets, but in reality, that's not what we do in the biotech lab. We use electricity.
Okay. We got a question here from Matthews. He's asking about the split process. Is the same, the split process is the same for bio, he's saying here bioinformatica, which is like bio IT.
Yeah, so bioinformatics is something that we use in the nucleic acids class. So it depends on how much into bioinformatics you want to go, but we touch into bioinformatics. So bioinformatics is, part of that would be generating those phylogenetic trees that I showed you when you're comparing, you know, viruses, one strain to another, or organisms to each other. So bioinformatics does a lot of those types of comparisons that I introduced you today. So understanding the basic molecule is definitely very important, but bioinformatics does a lot of sequence analysis.
Okay, I think Isadora, you wanted to ask something?
I want.
Okay.
You think no, but you know, if is there any way to change our DNA, something that can go wrong and change it in some way?
Well, so there's a couple of ways you can go with that question. So can you change your DNA? Well, yes, if you mutate it, right? So certain chemicals, UV light, radiation, they can cause mutations or changes in your DNA that you don't want, and that would be a negative effect. And that's in fact what happens to develop cancer, right? So mutations occur and cause the cells to behave badly. If you were born with a genetic mistake, there are techniques to correct those mistakes. That's part of genetic engineering. And there is a tool out there called CRISPR-Cas9 that is being used now to do things in animals. And it has a lot of promise, but it has not really been used in people so much yet, but probably in your lifetime, you will see a lot of CRISPR-Cas9. I know the Chinese scientists did announced that they were able to successfully introduce a change into some infants, but they were widely criticized for using it too soon before people really thought that it was safe enough to use. So in general, it's not being used in humans yet, but it will be, I'm sure. So yes, there are ways to introduce changes. And some of the other ways that they've used in the past have used viruses to introduce changes into the human DNA to try to correct genetic problems people were born with. But viruses have more problems than something like CRISPR-Cas9 could. So once they get a really good handle on how to use CRISPR-Cas9 effectively, I'm sure they'll be using it to correct mistakes that people were born with.
Okay, great.
Thanks.
Thank you, Isadora. Professor, we do have a question from Julia Magnarello, and she's asking, which area of research you most passionate for?
Well, I like microbiology. That's what I was trained in. I teach that class every semester. I teach students in microbiology, so I do love microbiology a lot. Many years ago, I did research on Lyme disease, but it's been quite a long time. Now I just teach more general, general topics, but I try to read literature and kind of keep up with what's going on in microbiology. So that's the part I like the most.
It's wonderful. So thank you very much, Professor. It was a very pleasure to have you here. It was such an honor, actually, to have you here talking to the students, and I'm 100% sure they enjoyed the opportunity. Now I'm going to open for Jetsa and Danielle. They can talk a little bit about this wonderful university that you are based at, and you are one of the directors. So I'll hand to Jetsa and Danielle.
Yes, hi. Hola.
Hi, Jetsa.
Hi.
You can open
Hello,
the camera if you want to.
Oh, I tried, but it's not working, but it's okay. I tried several times, so it's not working.
There you go, there you go. You can now.
Okay, let's see. All right. Goodness.
There you are.
Hola. So once again, I'm Hetza Torres from the International Student Program at Santiago Canyon College. We said SEC for short. So SEC is a fully accredited public college that is located in the city of Orange, California. Our college has 135 international students from 25 different countries, including Brazil. SEC offers 67 associate's degrees. These are excellent degrees that are in high demand, taught by excellent professional, highly qualified professors. So today you just learn about biotechnology and our wonderful professor, Dr. Foley. So for those of you who are not familiarized with U.S. community colleges or about associate's degree, so let me say that students attending SEC full-time for 2 years, they obtain an associate's degree. So an associate's degree means that you complete 2 years of your bachelor's degree. So in other words, when you graduate from SEC, you will transfer to a 4-year university to complete your final 2 years of your bachelor's degree. So 2 years at Santiago Canyon College plus 2 years at the university equals a bachelor's degree. So just a few highlights about SEC. I know we're limited on time here. So we're one of the newest community colleges in California. So with that, we have new modern facilities with state-of-the-art equipment. So Dr. Foley showing us, her lab earlier. And our college is a high-ranked school. In 2019, for example, we were voted number 3 best community college in California. And in 2017, we ranked number 13 among 50 community colleges in the US, so in the nation. So our class size, it's about 30 students in the classroom. Our tuition is very affordable. We have great support services for our students, and our school is very diverse. Our school values and appreciates diversity, and overall, our school environment is very friendly and it's safe. So we will be happy to welcome you all in the near future at our school. So any questions, I'm leaving my contact information with Diego, so please feel free to connect with me at any time. Thank you.
That's wonderful, Jetsa. Thank you very much. I just want to say one thing, Jetsa. For the British students, when they enrolled at the SEC and they don't want to do a bachelor course in any university, they want to stay in the industry, as Dr. Foley was saying. How is the process? Is it easy for students, alumni from SEC to get a job in the industry?
Well, we do have something that is called OPT. That stands for Optional Practical Training. And this means students, when they complete a higher education studies, in this case, it will be the associate degree that I just talked about, the 2 years. So students can work legally, okay, legally for 1 full year here in the US. So they apply for their Social Security, and they can apply everything that they are learning from that program. Let's say they come and they apply for the associate degrees in biotechnology. So students can apply what they learn in the classroom in real life. So working for 1 full year. So if those students, they can go back actually to and complete their final 2 years of the bachelor's degree if they wish to.
That's wonderful. Thank you, Jetsa. And it's an amazing opportunity, guys. SCC, it's a great college in US. If you want to have more information, I'm just going to pop up my email here. Most of you have my email already, but that's my email address. So if you want to know more about the opportunities that SCC got for you, please email me. Sorry, Danielle, do you want to say a few words?
Oh, sure. Hi, everyone. I'm Danielle. I'm the career education coach. Thank you so much for being here, Dr. Foley. That was a great presentation. You guys learned so much in just 1 hour. Imagine how far you can go going through the course and completing these certificates. And by the questions I saw and the interaction I saw, this is a room full of scientists. So I'm so excited to have you. My job is to just talk about biotech and talk to different students about where they see themselves fitting into biotech and how it can really relate to all of their career goals. So I have an amazing job, and I'm always here as a resource too. We also have an internship coordinator and job developer. As you progress through the program, we do help you find work, and this is work where you can make lots of money. And you can also do things if you want to maybe major in business, but you're interested in biotech. We can help you look at different positions on how you can hybridize a lot of your different interests. So this is a really great field. Our slogan here is, cure diseases, save lives, and protect the planet. So biotech can do it all.
That's wonderful. And Danielle, how much is the average of the salary of a biotech per year?
In California, right now, the average, so this is the average between having a high school diploma to a bachelor, so it's kind of in between. Yeah. It's about $96,000 in California right now a year.
Wow, that's pretty good.
It goes up the more you educate yourself, the fees or the price. The pay goes up.
Wonderful. Sorry, guys. Do you guys have any more questions for Professor Foley or Danielle or Jetsa? No? Okay. So that's great. So thank you very much, everyone, to join us today. Just one thing. I'm just going to talk in Portuguese very quick, guys, if you don't mind. Pessoal, essa foi a última mock class desse semestre. Agosto está recheada de mock classes. A gente está trazendo uma universidade da Finlândia para falar de engenharia sustentável. A gente está trazendo a SCC de novo para falar com vocês sobre business. Uma universidade de Barcelona para falar de business. E uma outra universidade da área de saúde, uma das mais antigas universidades da parte da saúde que prepara para a faculdade de medicina nos Estados Unidos para vocês em agosto. Então, fiquem atentos aí que em final de julho, começo de agosto, vamos começar a divulgação, ok? I just told them you guys gonna be with us again in August with business class. So thank you very much, guys.
We'll be happy to be back.
Amazing. Thank you very much. Thank you very much, guys. Have a nice day. Happy Monday.
Thank you. Muy obrigada.
De nada. Bye, bye.